Hello, my name is Ishita Bakshi, and I'll be explaining a research project conducted on the effect of gene mutations on the efficacy of a drug. In the medical field today, pharmaceutical drugs play a major role. They are essentially the use of isolated compounds and chemical synthesis to create stimulants that changes a person's physical or mental state. According to the Georgetown Health Policy Institute, over 66% of adults in the U.S. use prescription drugs, which is why it's important to understand the different effects they can produce. The first step in the investigation was to pick and research a commonly prescribed drug. In this demonstration, it will be celecoxib. The PharmGKB annotated drugs database was used to gather the following information. Celecoxib, also referred to as Celebrex, is primarily used to treat forms of arthritis. It's a type of medication known as a COX-2 inhibitor, an anti-inflammatory drug that targets COX-2, the enzyme responsible for inflammation. Next, the pharmacogenetics, which is the study of genomes in a drug's response, were researched. The main findings in this was that the drug is primarily metabolized in the liver by the CYP2C9 enzyme. So what exactly does this mean? From this, it was hypothesized that polymorphisms of the CYP2C9 gene could cause changes in the drug's response. Next, the pathways of the drug were studied to get an idea of what it directly interacts with, but there were no significant findings at this stage. At this point, it was inferred that deficiencies of the CYP2C9 gene could have an effect on the drug's response. So to confirm, clinical annotations of different celecoxib users were researched. The annotations had lots of information, including the exact gene in which the discrepancy occurred, the allele, which is an alternate form of a gene, the variant, which is the single nucleotide morphism, or SNP code, and a description of the abnormal drug reaction, or a phenotype. After reading through a handful of these annotations, I noted that the most common genetic discrepancy occurred in the CYP2C9 gene with the specific variant shown on the screen right now. The various annotations stated that the allele C was associated with things like poor metabolism, gastrointestinal bleeding, and a handful of other symptoms. Now that it was confirmed that a specific allele was related to celecoxib's abnormal responses, I wanted to take a look at the actual DNA sequence. To do so, the NCBI SNP database was found and used. The SNP code that was showed earlier on the screen was researched and looked up. And here's what it provided. In this first red circle, you can see the actual part of the DNA coding sequence where the mutation occurred. Now this next piece of information shed a lot of light. In the next red circle, you can see how the mutation changed this three-letter sequence. So what exactly are these three letters, and what do they have to do with anything? Let's take a look at genetic coding. The language of mRNA in genetic coding and DNA replication is called genetic coding. It's read in three-letter sequences known as codons. Each codon codes for a specific amino acid to be added to this long chain called a polypeptide. These polypeptide chains are what make up proteins, and the properties of proteins are determined by the amino acids joined together. Let's take a look back at the allele change now. The change in the codon resulted in a different amino acid to be added to the polypeptide chain, as seen on the right. All of this information was charted and analyzed. After piecing together all of this information, it was concluded that for celecoxib, different genetic mutations in the CYP2C9 gene can alter how the enzyme functions and metabolizes the drug. All in all, different types of genetic mutations can cause changes in a polypeptide chain and create a different reaction from the drug because of the alteration in the protein function. To further look into why proteins change, you could look at the different characteristics of the amino acids and what makes them different. Lastly, we have some post-procedures observations and questions on topics like the different types of mutations and the differences between amino acids. Here that we're excited, and thank you for tuning in.